Hello, everybody. I am Ken Nicholson. I am the Associate Professor and Chair of the Department of Chemistry at Northeastern Illinois University in Chicago. I am involved in an interdisciplinary environmental science program. And this presentation will highlight some of the things that we did this past fall in one of our environmental science programs. I did this with two of my colleagues, Greg Anderson and John Kasmer, and they are co-authors on this presentation, but are not in attendance today. So we have, in the environmental science program, is interdisciplinary by design. It contains members from many different STEM disciplines, such as biology, chemistry, earth science, and physics, and we regularly co-teach all the courses in environmental science. And so we can get different perspectives and points of view. In this particular course, Field Methods in Environmental Science, which is NV301, this past time it was taught by 3 faculty members, one from biology, one from chemistry, myself, and one from physics. And we used a modular approach to teach this course. It started off led by the biology faculty member with a local survey of water quality and ecology, where goals were of learning statistics, the use of field probes, field notebooks, writing in the discipline, some standard operating procedures of water collection, soil collection, sampling, storage were introduced and explored by the students. And we did this in the framework of looking at our local water quality around our campus at many different neighboring sites. The next part of the course involved testing of water, an analysis of water samples for persistent polyfluorinated alkyl substances. These were mentioned in the previous talk as being an ongoing problem, and it has been becoming a bigger problem around the Lake Michigan watershed area. I led this part of this course, and what we did was we collected several different samples and sample types, both drinking water and river samples around Lake Michigan. We learned how to use follow published EPA methods and modify them to meet the limitations of our equipment and what we have available on site at NEIU. We learned techniques such as solid phase microextraction and high performance liquid chromatography. The course closed by looking at how air quality can be impacted by Lake Michigan. And so the students became familiar with using and importing data from EPA data sources. They learned how to program in R and create these data visualizations. Next slide, please. And so this map is a map of where polyfluoroalkyl substances, PFAS contamination has been found around Lake Michigan. And all the blue dots represent where it has been found above the limit set by the EPA. And so we decided to sample several of these areas, and these areas are highlighted by the red circles. So in Western Michigan, there is Hamilton, Michigan. That's the northeast part of this map. New Buffalo, sort of the southern part, right on the edge of the Indiana-Michigan border. The red circle that doesn't have a blue dot right inside of it is our campus. And then the Fox River. Next slide. And so we used EPA method 537.1 to do this work. And so to establish a method, the students needed to learn how to establish a method and develop a method in chromatography. To do this, we purchased and used PFAS compounds on the list to spike the sample, basically a deionized water sample with things that we bought and just purposely put it in there to see how we could possibly get separation of these compounds. And so I'd like to highlight some of the features, some of the peaks with my cursor. I hope that you can see that, particularly the ones around 6 and 8 minutes. Those are the ones that we assign to the major components of the spike. there's the spiked reagents are not perfectly pure, so you'll find some impurity peaks. And so just to give an example, and since we have only 5 minutes, we found what we believe to be possibly some PFAS contamination in the Hamilton sample, with peaks somewhere around 6 minutes, and also in the McHenry River water sample around 6 and 8 minutes respectively. These are in the range that we established with our spike sample that we would expect to see PFAS compounds. We did some elimination of some other compounds and so on, but we can't, with our equipment, we had to modify the EPA method, so we are not able to get the mass spec, but this provides some data for us to propose a grant and get funding to purchase equipment that would be more suitable for this project or easier to use. Sample closed. Our course closed with Lake Michigan influenced air quality. And so this was one of the generated plots by the students that showed, how when ozone increases, there's a decrease in nitric oxide, NO2, and that they're somehow interconnected with one another. And we have a chemical explanation of that through reactive mechanisms and so on. We also found doing some wind studies that and that the wind, lake breezes coming off the lake impact both the ozone amounts that we might find, and the NO2 amounts. So I do appreciate your attention, and I look forward to the next 3 lightning talks.